All right, this is lecture 14 on gene finding. We're going to explore today Markov models and related hidden Markov models. But before we get into that, I wanted to talk about one feature of R. Um, it's not particularly related to gene finding in any way, but it has cropped up actually subtly in the lecture last class, I believe, and it'll crop up here and there when you look online for solutions and packages. As some programmers use this technique called object-oriented programming. Um, it's far beyond the scope of this course to give a complete exposition of this concept, but the basic idea is pretty straightforward. And it's actually a very powerful paradigm that really helps to keep your code in a very organized way. Um, Now, in R, it's actually a bit strange because there's multiple ways of doing object-oriented programming. I think that's probably because, and I'm not an expert here, the object orientation was sort of added on after the fact, so a posteriori, and it wasn't really built into the language at the beginning. And so perhaps there were competing um, views on how to implement object-oriented programming in the language. Regardless, there's this S3, which a lot of people know at some level. What I mean by that is it's usually people's first introduction to object orientation in R. And then S4, which is slightly more difficult than S3. And then you get into these RS levels, R5, R6. And to be honest, I don't know R6. I haven't really. I would like to get to know it, but I haven't had the opportunity to date. We're going to look at S4 today. I think it's a nice compromise between the unstructured approaches of S3 and the more formal approaches of the R5. Okay, so the basic concept are as follows. OO programming begins with the concept of a class, which something that you already basically, you have a handle on already, that there's a class of numeric or character or logical, etc., lists, data frames, tibbles. Um, you can actually make new classes on demand, and it's a little bit more complicated, the difference between type and class in R, but this is good enough for our purposes. So we can make classes on demand. And then we can make objects um, or variables, really, of those classes. So for example, we could think of a genome as a class, right? So just kind of close your eyes and visualize this. And so the idea of object-oriented programming is that you treat this genome as an object that sits there in space, right? You kind of envision what a genome, and there would be a whole bunch of um, objects, one for each genome that you know. For example, there would be this genome object for yeast, one for human, one for rat, etc. Right? So the objects are instantiations of the class, right? So it's like you can say, well, we have numerics, and 1.3 is one instantiation of a numeric, and 7.4 is one example of a numeric. Just like the yeast genome is one example of the genome class. Now in R, we can use this function setClass, which I'm going to show you in a few minutes. It's from the R base language, so there's no fancy packages here like the tidyverse. And the setClass has both required arguments and optional arguments when you're setting up a class, and we'll get to that in 1 second. But the real power, or one of the real powers of object orientation, is that this genome object can have many properties or attributes. In R, they're stored in what are called slots. So now when you visualize that yeast genome, you can imagine there's a name associated with that yeast genome. There's annotations, like we looked at last class. There are NCBI identifiers, all sorts of information about that yeast genome belong in that class. In fact, of course, that also includes the DNA of the chromosomes itself. So what object orientation allows us to do is package all those things together. that are all related to the yeast genome. So think of it, before we go on to the concrete example, um, a more pedestrian example would be to think of a house as a class. And so I own a house, so I have one instantiation of that class house. And then every house has properties like how many floors. That would be one slot. So my house has 3 floors. Uh, Brick versus vinyl, right? How many bathrooms, the size of the space, the owner of the house. All of these things are attributes, and they belong all together um, in that object called a house. And in general, object orientation can also have superclasses. So you might think of a house as being a subclass of a building. So buildings can be houses, but they could also be skyscrapers, right? So you could have different classes that are all contained under a superclass called building for the different kinds of buildings we have: houses, greenhouses, sheds, etc. And within the object or the class, sorry, within the class house, we could also have subclasses. For example, you might describe, let's say, a bathroom by its attributes in terms of, let's say, floor heating, or a sink, or a shower versus a bath, etc. And so, because bathrooms could be maybe in different kinds of buildings, it would make sense to collect those objects, those attributes of a house, into one subclass that might be used in other types of buildings, right? Okay, so that's a little bit abstract, but there is another property of object orientation that has to do with that, defining methods on the objects, and we won't talk about that today. Okay, so here's an example that comes really organically out of the course. And last class, it was starting to get a bit confusing having all of these different annotations about yeast, right? So we can create an object called genome that's going to package all those together. So this is just an arbitrary name. Here I've called my class genome. This is the set class function that I promised to show, okay? Now the first is the actual name of the class. So this would be like saying numeric, or character, or logical. In this case here, our name is going to be genome. And usually these match, right? These match together. It's a little bit confusing why you need to repeat that, but just do it. So there is a convention often in object orientation that new classes have a capital letter. It's not necessary, but it's classic. Now, here are the slots for our genome that store attributes about our genome, and they're pretty intuitive. Organism name, in our case, will be something like Saccharomyces cerevisiae. The chromosomes themselves, and we could reuse the DNA string set that we saw from the BioString packages last class. The annotations in chromosomes names and Go annotations, well, we can make those data frames, or tibbles if we wanted to, but here we make them data frames. Okay, so that's our slots, and of course we could add more slots, more attributes to our class genome, but that's good enough for now. And oops, scrolling down, I need to get off of that pencil. And then there's two other parts of a class. One of them is the prototype. I think it's necessary. It basically says how to instantiate a new object of class genome. So what is its default value? If we don't specify an organism name, chromosomes, these other parameters, we're basically saying here what it should be given by default. So our organism name will be the empty string, the chromosomes will be an empty DNA string set, and the rest of the annotations will just be empty data frames. It's just basically saying, okay, if you don't give me any information, this is what I'm going to pass you back. And there's one final thing here, the validity check, which we won't go into here. Basically, the validity function here is just empty for us. Basically, it doesn't do anything. It doesn't do anything. You can use the validity to check that the parameters that you pass to the function, to the class, when you're instantiating an object, this validity function can check that everything is correct, everything makes sense, that you're not passing a number for chromosomes, that you're passing actually a set of DNA strings or something that can be coerced into a set of DNA strings, that the organism name isn't illogical, et cetera, et cetera. So you can check. It just basically is a quality control mechanism. Okay, so now that we have our set class, we can really go to, that's it. Now we can go to town. So here I'm going to call one, I'm going to create an object, a genome object called bland, because I don't pass it any parameters here. And so what we see if we print out bland is basically that the organism name is empty, the DNA string set is empty, and the data frames are all 0 rows, 0 columns, right? And okay, so we created, that is, bland is now an object of class genome. Now it's boring, but it's bland. But I can access now, and this is what comes up, and why I need to show a little bit about object orientation in R, is that you'll see sometimes the use of the @ operator instead of the $sign. And this is a bit confusing to both new and experienced R users. So normally we would say bland $sign organism name to recuperate a name of the object bland. But here, with the @ operator, allows us to sort of reach into that object called bland and pull out the organism name. So this is allowing us to access the slots of the object. So if I ask for blanded organism name, it just returns the empty string. Same thing for if I ask for the chromosomes, it just gives me back an empty DNA string set object. Okay, so you know, if we go back to our house example here, it would be if we had a class house with all sorts of attributes that I listed before, you would say something like, you know, instantiate, sorry, let me scroll back up. You know, my house is set equal to the host, you know, object. And then, you know, it might be like host at address is the address of my house, and host, my host at, you know, floor space might return a numeric, which is the square meters or something like that, right? Okay. So why is this important? Well, I think here's a beautiful example, a second example of creating a genome object of class genome. And you know, basically remember last class that we started to collect all this information about each genome, and we can load them up individually. But aren't they kind of floating around in your environment? You know, we have to remember that ANO is the annotations, SC is the actual chromosome sequence. You know, it's really kind of a challenge to keep all of that information in your head, right? And if we had multiple genomes, it would be really impossible, right? SC is short for Saccharomyces cerevisiae. You just lose data. But now what we can do is in our genome object, we can create a genome called S. cerevisiae from the class genome, and we set the organism name appropriately. We pass the sequences to chromosomes, right? That's a DNA string set. We pass the names from SC meta, the annotations from ANO, and go to go, okay? Okay, so now if I ask for the slot organism name, it returns, not surprisingly, Saccharomyces cerevisiae. That's nice. And I ask for the annotations, I can see that's the Tibble that we wrangled into our last class. I can ask for the length of chromosome 3. So here I've asked for the slot that contains the chromosomes, and that's a DNA string set. So if I ask for the third element in that list, the contents of the third element, because it's a double square bracket, I get back the number of base pairs in chromosome 3. And in fact, now, and this is really the powerful thing, is that if I write this object, S. cerevisiae, this object to the file into my, my files in the project in RStudio Cloud, it's there for good. So when I load this guy in the future, everything is packaged together, and it's really easy to find by just referring to the slots of the different pieces of information. Okay, yeah, as I mentioned briefly, there are another aspect of object orientation is the fact that we have functions that operate on our objects. We won't go into that. But for example, we might take that function I wrote last class called MyScan that looks for a binding site in a chromosome. And you know, MyScan, you know, again, you know, right now it's sitting there in your environment as a function. It's not really packaged together with the whole genome object. But you know, MyScan doesn't just work on a yeast genome; it works on a human genome. So you could put this inside the class of genome, and now MyScan becomes a method, a function that's specific to genomes. So whenever you create a genome, it has its special function for looking for binding sites, and that's really maybe. Well, I hope that you start to get some intuition as to why object orientation is a powerful programming paradigm, and most modern languages have some notion of object orientation. Now we'll return back to transcription factor binding sites and talk about a few remaining concepts that we left open at the end of last lecture. In particular, we didn't really cover too very much about how we evaluate the performance of our model, and partly that's because it's actually very difficult to evaluate transcription factor binding site predictions. So let's talk about that a little bit now. So at one point we built this log odds ratio of the observed motif at a position in the chromosome versus a background model, right? So we had the log of the P of X divided by the background across the chromosome. Okay, and this was for 4 different TFs. I plotted this out, and yeah, hopefully you can see the x-axis pretty well, but this is 0, 0, 0, and 0. So 0 means in that log odds model that it's equally likely that this was generated by random, the background, the denominator, as compared to being generated by that actually being a binding motif for the transcription factor, this P at X, right? So it's equal. So anything greater than 0, there's more evidence for it coming as, there's more evidence that it's a binding site than that it was generated by random. So we could, for example, draw our magic line in the sand here at 0. That would make kind of like an intuitive sense that we're saying, okay, anybody to the right of 0 will predict as being a binding site, and anything to the left of 0 would be designated as not a binding site. And of course you can see here already that depending on the transcription factor, this guy here, I don't remember which ones were which, I should have labeled these better, but there's a small number of sites that we would predict to be binding sites, and there's very few for this guy, and probably quite a bit more for this guy here and this guy. So the number that you would predict as being true binding sites is actually dependent on the motif itself, which is kind of a bit something to think about, at least what that means. But you know, we might say, oh, 0 is not strict enough, then we should shift this line over to say, I don't know, 5, right? So there has to be not just a little bit more evidence than that the motif, there has to be significantly more evidence that the site corresponds to an actual motif than just background. You know, imagine, should you accept, for example, something that's 0.1 as being a true site? So sometimes you see that we add an epsilon to shift this to the right to make us it's more conservative, right? We make fewer calls. Okay, so no matter where we set our line, we have this issue then of how do we evaluate the classifications. And so what we're doing here is a classifier. So what we're saying is for each position in the chromosome, we're labeling it as either a binding site or not a binding site, right? So now that whether we label it as binding site or not binding site is determined by where we set that point along the x-axis, right? So anything to the right is we predict as being a positive, and anything to the left we would say is not going to be a hit. That's our predictions, right? That's our classifications. It's not reality. In general, we don't know which are true or false, right? We're trying to learn that. Now, we need different definitions here that come up over and over again. And this is, again, the rudimentary ideas underlying classification and machine learning. So the first idea is that of a true positive. So if we predict a position in the chromosome, right? So here's our chromosome, and we're looking at this window here for our motif, right? And if we say, okay, this guy scores 5, then because of our line in the sand, right, for this distribution, if that's 0 there, that means that we might use, if our line was 0, we might say, okay, 5 is bigger than 0, so we'll predict it to be positive. Now, if it turns out that we can validate that this is truly a binding site at that position, we'll call it a true positive, right? So our prediction was positive, meaning this site was actually a binding site, and it turned out to be actually a binding site in reality. A true negative means that we would be a site that we predict, let's say maybe it scores -3, and so it's down here, and we say, nope, that's not a binding site. And it turns out that we could validate that, in fact, it's not a binding site. So when I say validate here, I mean that we would go in and say do an experiment like CellX or something, or we'd have to do some actual lab work to show that that in fact doesn't bind the transcription factor, right? And that's admittedly very hard to prove a negative, right? I mean, think about that, right? In some ways, proving in the lab that something is a true positive might be easier because you would simply try to express the transcription factor and then detect when it binds in that location, etc. But if you're trying to prove it's never binding a transcription factor, that's hard, right? Because how do you know you've covered all the conditions of the cell that might bring that transcription factor to that spot, right? And it might not just be that transcription factor, it might be an ensemble of cofactors and RNA molecules, et cetera, that help it bind or facilitate it binding. And so, how would you in the lab really create that biological reality that might actually, witness binding of the transcription factor to that place? So it's not really easy to prove something is not a binding site. Often the arguments are more like, this site, the first gene is way, thousands of base pairs downstream. So it's like in the middle of nowhere. This guy here is like nowhere, and this guy here that's positive. Oh no, the negative guy, right? So this is the true negative we're talking about. So, this guy here is so far away from the next gene that it likely it's not involved in its transcription. Bit philosophic. And then, okay, so then on the other hand, we have concepts like the false positive, which means that we predicted that some site here we predict it to be a positive, so maybe its score was 7. Okay, so because its score was 7 in that distribution, it exceeds our line in the sand was 0, so we predict it to be a positive, but it turns out that it's not, right? So that, again, if you could prove in the lab that it's not a binding site, then this would be called a false positive. So that's not something we want, right? So we called something positive that's not. Lastly, a false negative is a position that we predict. So we might say, okay, this guy had a low score, -11, way down here. We say, no, it's not a binding site, so we say it's negative, right? So, but it's false negative because we could validate that in fact it is a binding site for the transcription factors. So these two guys are, of course, not what we want. We want to maximize our true positive and true negatives, right? And if we were to just have true positives and true negatives with our classifier, then we'd be in business. So this kind of spells it out a little bit more detail, and um, I suggest that maybe you study this figure a little bit uh, on your own too. But I'll go through it quickly here to try and make sure everybody understands. So this distribution here again is the distribution of scores, right? And this might come from log our log odds model, right? P of x divided by the background, right? Okay, sorry, the background, okay. And so we might decide to make our cutoff here, which is 0, okay? So any scores above 0 we're going to call a positive, and anything to the left is going to be a negative. Now, I put these x's in here because each x corresponds to a position along the chromosome. Now, I didn't draw arrows for every gray node. For every green x, I did draw an arrow. But conceptually, every one of these x corresponds to some position along the chromosome, and that's the score. So this guy here that has a score of, say, minus 15, might correspond to this position on the chromosome, and that position had a score of minus 15 by our model, right? Okay. Now, in fact here, the x's that are green are true positives. Now, you can see, right, that already that they're kind of scattered. There's maybe enriched up here in this upper tail, which we might expect if our model has any value, but there are some green x's below our cutoff value. In fact, you could see right away that no matter where I put this cutoff value, except maybe way up here, if that's a green guy, if I put it way up here, I'd get this guy right, but I'd get all those other green guys wrong. And that's the crux, right, of where we're going with this. And if I put this cutoff value all the way down here, well, I would miss all of the true positives, but I wouldn't incur, I would be calling every one of these guys negative. And so, yeah, I'd miss the positives, but I'd get the negatives. Okay, so, but one thing at a time. Each point, like this guy here, maps down to some position there, and it's a true positive, right? Okay, so the point here, the last thing here is that a point like this guy, okay, if we follow it down as here, it's a true positive, and he's to the right, or she's to the right of this cutoff here, and that means that we predicted it as being positive, so we got it right, right? Okay, we got it right. This guy is also to the right of this line. It's green. It's a true positive. And what? Whoops. I made a mistake. Okay, I made a mistake. So this guy here, I think I got confused because I think that's green. Let me annotate this quickly. It's green, okay, that point. So it is actually a true positive, and because it's to the right of this line, when I follow this arrow down, it should be, I put a green there. And because that line, it's to the right of the line, in fact, this is a true positive, right? Okay, I'm sorry I made that mistake. All right, okay. Now, this point here is also green, so if I follow it down, it's green, so it's a true positive. These guys, this guy here is just to the right of the line, it's true positive, and this guy here goes all the way there, so that's a true positive too. So we have some true positives. What do we have any false positives? Of course, right? This guy here is gray. This guy here is gray, and if I come down, it's not a positive, but he would be predicted by us to be a positive because he's to the right of that line, and so that's a false positive. This guy here is our first false negative, so if I trace this guy back, I see it's coming from a point up here. Now, it's actually green, so it's actually a true binding site, but it's to the left of this line, and so we missed it. So it's a false negative. We predict it as being negative because it's on this side of the line, but falsely so because it's actually a positive, right? So that's bad news. And the last concept we need is a false, well, we have false positives, right? False negatives, true positives, and true negatives. There's lots of true negatives, right? All of the gray guys here are all true negatives. So for example, this point here comes in here, it's a negative, and we predict it as being negative, so that's a true negative. Okay. Now the last caveat here that I want to mention is, maybe, I hope that you can read that. This is the background. This pizza thing doesn't work all that great. That's my background distribution, right? Okay. So the last thing I want to point out here is that we start off with just partial knowledge of the binding sites. Maybe we have 6 of them. And there's more than 6 green X's here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Who knows? Maybe there's a couple hundred binding sites that are for that transcription factor, of which we know 6. And those 6 were used to parameterize our model, right? To build our model, to build the position weight matrix. However, some of these guys, and I denoted them here in dark green versus light green. So these guys with the underscore here, just these T's, this true guys with the underscore, they correspond to the known binding sites. The dark green guys, this guy, this guy, this guy, this guy, and this guy, we didn't know them beforehand, so they're new discoveries, right? We've used some information to predict the rest of the binding sites in the genome. And if you look closely, if you blow this up, you'll see that there's dark green and light green. I could have maybe perhaps made it a bit more distinctive. I apologize for that. But the dark greens were not part of the data that was used to parameterize our model. Why is that important? Well, it turns out that is extremely important and a flaw in my approach here, in fact. Because when we evaluate the accuracy of this model, the performance, we're going to be we're actually including guys that we used to build the model, and generally that's a concept that we want to avoid. If you've read the chapter, the reading for this lecture, you know that they've already started to talk about the importance of separating the learning phase from the testing and validation phases. Okay. And the basic idea is that whatever data you use to learn your model or parameterize your model, and in this case here again, that's our 6 binding sites, it shouldn't be then used in the evaluation of our model. That turns out to never be fair. Okay, we'll come back to this concept in more detail. Okay, so these are your classic measures of performance, and I have trouble understanding and remembering these all the time. So if you have a challenge with memorizing these or understanding what they are, you're not alone. I always have to go back and look them up and refresh my memory as to what they mean. But let's focus a little bit on the intuition and I'll go back to our previous example. So the sensitivity is the number of true positives divided by the number of true positives plus false negatives. Okay, so let's go back here and take a look. Now, a true positive means a green guy to the right of our line, and a false negative is a green guy to the left of our line, right? So, for example, here, if I come in, this is a green guy. He comes down here, and it's a false negative, right? So we should have predicted it positive, but we predicted it negative falsely. So right now, our sensitivity is basically counting the true positive, which is 1, 2. Well, wait, I should be able to zoom a bit. 1, 2, 3, 4, 5, 6. I have 6 true positives. And how many false negatives do I have? 1, 2, 3, 4, 5, 6. So my sensitivity is actually a half. You can validate that on your own time, but I believe my arithmetic is correct. I have 6 true positives and I have 6 false negatives, so my sensitivity is 6 divided by 12. Now, what's the intuition of sensitivity? Here's the best way to do it, I think, to understand sensitivity. If I move this bar all the way to over here, to this line, okay? Let's see if I can, if I move this bar all the way over to here, the very extreme, okay? So I've moved this cutoff down here. My true positives are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 12. My false negatives are 0. So what is my sensitivity? It's perfect. It's 1. Sensitivity goes from 0 to 1. Okay, so I have perfect sensitivity. I get all of the true binding sites predicted by my model. Now already you're saying, but that's crazy because all those negatives, that's not sensitivity. Let's not get ahead of ourselves. If I move this bar all the way over here to say some point down there, right? Now, every one of the green guys is a false negative, and my true positives are 0, so my sensitivity is 0, right? Over here, my sensitivity is 1, right? So my sensitivity is awesome if I'm willing to absorb all these false positives. And if I move it over here, my sensitivity really sucks because everybody is missed. Okay, so sensitivity alone isn't so meaningful, right? Because it seems like something else. But the idea is that, how you think of your algorithm as being sensitive if it comes upon in the forest, as it's walking through the forest, it comes upon a cute little puppy dog that is actually a true binding site. It's sensitive enough to recognize it as a cute little puppy dog, put it in his arm and take it home, right? An unsensitive classifier, as it walks through the forest, it meets a little cute puppy dog and it just leaves it there as a negative, right? I think that really brings it to life. Now, specificity is different. Here, it's the number of true negatives divided by the number of true negatives plus false positives, right? So a true negative is the gray guys, right, in our figure. And the false positives are guys to the gray guys to the right of our line. And sorry, the true negatives are the gray guys to the left of our line, right? So now this is just, now you can, in your head, you can just delete the cute little green puppy dogs. You're only looking at the gray guys now. See, the sensitivity only looked at the true positives, like the green guys, and now the specificity only looks at the gray guys. So if my bar is here, and my specificity is going to be the number of, sorry, let me go back here, the number of true negatives, right? So where are my true negatives? That's all the gray guys down here, and there's an awful lot of them. And where in the second, now, so that's the numerator, right? And now I need to know the false positives. The false positives are all of these gray guys over here to the right of my line. So it's going to be the ratio of all of these guys restricted to gray divided by all of these guys together restricted to gray. Okay, so these are my mistakes, the gray guys in here, right? So again, okay, so here I can't count all these points, but let's suppose this is 1,000 points, and this is, I say, 100 gray points, then my specificity is going to be 1,000 divided by 1,000 plus 100, so it's going to be something like 10 11s, right? So it's going to be pretty close to perfect just because there's so many negatives. Now, my specificity, if I think of my specificity now, I come in. I don't understand why my little pen stops working sometimes, but if I go ahead, come back, nope. Okay, so it stopped working here. So if I put my specificity, my cutoff here, what happens to my specificity? So I don't have any true negatives, right? So my numerator is 0. And everything, all the gray points are false positives, right? Because if my line is down here, all these gray points are predicted correctly. So my specificity is 0. So you have to imagine a 0 right here under specificity. Now my specificity on this end, if I move the cutoff over here, what happens is that all these gray points are correctly predicted as true negatives. So my numerator is all the gray points. And I have no false positives, right? because if my line is here, there's no gray point predicted incorrectly. And so what that means is this is now equal to 0. So my specificity is just basically all the gray points divided by all the gray points, which is 1. So specificity is sort of counterpositive to the sensitivity. Specificity is 0 down here and 1 up here. Okay. So now, so alone, like they're counterpositive to each other, but they're not very useful. It's accuracy, which is a combination of sensitivity and specificity. And it's kind of intuitive, right? It's the number of true positives and true negatives your classifier picks out divided by all points. This is just basically all possibilities. So when we go back to here, what that denominator is all these points. And then what you're saying is, how many true positives do I have? That's these green guys here. And how many true negatives do I have, which are all the gray points there? Now, if my cutoff was, okay, so here's the point. Do I have perfect accuracy? No, because I have mistakes, these green points down here, and these gray points up here. So I don't have perfect accuracy. Is there a place I can put my cutoff where I have perfect accuracy? So maybe you want to stop the video and just think about that a little bit, shift this around. The answer is, of course, no. Anywhere I put this line, I'm going to incur some false positives or false negatives. So there is no way in this simple scheme, looking at this distribution of scores from the motifs, to perfectly classify binding sites. That's often the case in classifiers. It's too much to expect that you can ever perfectly predict. What you're hoping to do is predict better than random. We can come back to that later. Okay, now the last thing I wanted to say here is that, and I hinted on this before, is that if you're trying to actually validate whether sites are true positives or true negatives, that's pretty difficult and maybe not symmetric. Like I said before, it's maybe easier to identify experimentally in the lab whether something is positively a transcription factor binding site, because you just need to get the cell conditions correct to show that it actually does work. True negatives are hard to validate because under some cellular condition, maybe there's a set of transcription factors and cofactors that do actually upregulate the gene. In general, in practice, what people do is they argue that if the transcription factor binding site is too far from a gene, or up too far upstream, etc., then it's probably not a real binding site. That's difficult. That may work in things like yeast, but in human, that may not be as safe to say. The transcription factor binding site prediction in general is a really difficult problem to validate. Okay, and I want to come back to this issue here, is that some of the motifs that were used to train our model, okay, and that was, if I go back to these slides, these guys down here, these binding sites that we used to train our model were used in the evaluation of its performance, okay? So concretely, I don't want to make you dizzy here, but so these, any true that had an underscore is actually part of the data that was used to parameterize our model, to create our position weight matrix. These dark green T's were not used. We didn't know that they were true positives, okay? So when we calculated our accuracy, specificity, and sensitivity, in fact, too, we didn't consider that some of these were actually part of the training data. We probably should have removed them from our calculation of how accurate the model is. It's not really very safe for you learn a model, and you might be sort of overtraining, what's called overtraining the model, overtraining. And when you overtrain, basically what it's done is just memorize the position of the correct true positives that it's seen already. So okay, let's just keep this intuitive. If you built a model like this and it was only capable of identifying those motifs that it was learnt on, that's not very interesting, right? You want to know whether that model has predictive capacity to classify new sites in the genome that are either binding sites or not, so either as true positives or true negatives. And like I said, the required reading for this week starts to touch on this issue, but we'll come back to it. And I want to start this really early, that you always understand that training your model. So when you build that position weight matrix, that's a separate event from how you validate your model. Okay. Now, one simple approach. This is really a thought experiment for you, a reflection point for the class. Is one simple approach to kind of fix this problem right now is that we would only use 3 of 6, for example, of our known binding sites. And then we would evaluate on the other remaining 3 after we built our model. So why is this not quite correct? And I'll leave that to you as a little puzzle, okay? So just to make sure that you understand, if we go back to here, we would take away, say, randomly choose just 3 of these 6, build our model, run it across the genome, do this analysis, and then we would see if it picked up the 4, 5, and 6 if we trained on 1, 2, and 3, for example. It's not quite correct. I'll leave it to you to figure out why. Okay, so now we're going to switch to Markov models and bring these concepts of model and performance and evaluation into gene finding, a second problem that's related to binding site prediction, but a little bit more robust. I put some points of reflection here. I should have really added to that this example from 2 slides ago about why that scheme of selecting only 3 of 6 isn't quite right still. Okay, but I'm not going to come back to these slides now, and I'm going to switch over to some handwritten notes. Okay. All right, so we're going to start learning a really powerful technique from machine learning called hidden Markov models. And hidden Markov models are based on a very powerful mathematical concept called the Markov model. Okay, so in fact, I would say a good fraction of all computational biology software out there is somehow based on a Markov model. And we're going to come back to this concept many times throughout the course. Okay, so a Markov model, or MM, consists of 3 things. Firstly, we have a set of states or nodes. That's these round things, A, C, G, and T, for example, but it could be 1 state or 10 states or 1,000,000 states. In this case, we have 4 states. And it consists of transition probabilities between states. That's these directed arcs that leave a state A and enter a state C. And in this case here, you'll notice that every possible transition between 2 states is represented, from C to G or from T to A. And in fact, even you have self loops, which are from A to itself. Okay, so that's the second thing. Now the third thing is the probabilities for this, what we call the probabilities for the starting states. And I've listed these down here. Okay, down there. And basically these initial probabilities dictate what the probability is that we start in state A or C or G or T. In this case here, the probability is 1/4 that I start in state A and 3/4 that I start in state T. And there is no chance that I start in states C or G. Okay. Now, these guys, these 4 values, have to sum to 1 because that's going to be a probability distribution. In essence, what we're saying is that we have to list all the possible different outcomes. In this case here, there's 4 different possible outcomes, and 2 of which have 0% chance of occurring. All right. Okay, and likewise, for the transition probabilities between states, they have to sum to 1 for each outgoing arc. So I have a slide to show this a little bit better. The next slide. What's going on? Yeah. Okay. So in this case here, let's focus on A. Now, there are 4 arcs that leave A. A to G, it has a probability of 0.2. A to C, which has a probability of 0.2. And the last one is A to T, which has a probability of 0.1. 0.2 plus 0.1 plus 0.2 is 0.5. And not surprisingly then, the self-arc for A staying in the same state A has probability 0.5. Okay? So all of the outgoing arcs from a state have to sum to 1. For the same reason that the initial probabilities, for example, these guys down here, have to sum to 1. All events have to be listed. Some events may have 0 probability though. For example, G has 0 probability of transiting to A, 0 probability of transiting to C, and 90% chance of staying in state G, and a 1% chance of switching to T. You can verify for yourself that state T has a quarter probability for all 4 of the possible transitions. Okay, so the idea here is that you can think of yourself as being a player in this Markov model, and you're dropped onto a node at the beginning of the game according to these probabilities. So you know that you're not going to parachute to C nor parachute to G. You might parachute to A, but most likely you'll parachute to T with 75%. So we would roll the dice and we would start in state T. And now we move, so you can imagine yourself standing on state T, and you roll the die, and with 25% chance, you'll go to C. With 25% chance, you'll walk over to A. With 25% chance, you'll walk over to G. And with 25% chance, you'll just stay exactly where you are. And you'll keep repeating that. So let's suppose I rolled the die and I switched to A. Now I would roll the dice again, but according to these probabilities, they're the ones that dictate where I go next. So I might stay here with 50% probability. And then maybe I roll the dice again and I end up going back to T or C or A, then G, then things. So I'm going to walk through this Markov model in a probabilistic way. Okay. Sometimes putting these values on the arcs is a bit awkward, and so we re-express the transition probabilities in this transition matrix. So the way we read this is from A to A is 0.5, from A to C is 0.2, from A to G is 0.2, etc. So we should probably say something like from to, right? Okay? And you can verify for yourself that this matrix reflects the same probabilities as this diagram or this graph. Okay? And sometimes we use pi sub A, pi sub C, pi sub G, pi sub T to represent the initial probabilities as a shorthand. Okay? All right? Okay, so go through this a bit more slowly. I was in state C. I throw the dice. Suppose my random number is 0.5. Okay, so now I look at my vector down here, and from 0 to 0.3, from here to here, right? C to A. If my random number is between 0 and 0.3, I transit from C to A. But if it's between 0.3 and 0.6, up here, I transit to G. Notice here that the probability of C is 0, so it doesn't show up in this schema here. My random number is 0.5, so I actually transit from C all the way over here to G. Okay, and now this one, I just now go back to step 1 and repeat the process. So for example, now I'm in state G, and I throw the die, and I get back a number 0.2. If I look at the vector of down here for G, my number from 0 to 0.9 is an A, so there's a high probability I transit to A, and that's my number suggests that, and only a 1% chance, or 10% chance I would have switched to a T, which means now that I will go from here to here. Now I'm in state A, okay, and I'm no longer in state G, and I can update that. Whoops, my eraser, right, okay, come back. That's state G. I've left state G, and I'm in state A. And you'll notice that as I walked along, I basically went from state T to state C to state G to state A. And so I'm generating a sequence by walking through this Markov model over time. And if I went on arbitrarily long, that random walk would produce a longer sequence, right? For as long as I wanted to. I could simulate an entire chromosome, for example. Okay. Hopefully that's a Markov model. It's a really simple idea. It's been around, I think, since the 1800s. And it's really powerful. It's really, really powerful because it allows you to generate very complicated statistical distributions. Okay, so let's play some games. Start. Here's a simple Markov model. I have two states, A and B. And these are my transition probabilities. So if I'm in state A, 90% I stay in A. I go to a B. If I'm in B, I stay a B. My initial probability is 1, and my for state A, and there's no probability I start in B. So you should stop the video now and answer the question, what happens in this, what are the kinds of sequences that this Markov model would generate, right? State sequences. For example, would you find a sequence that looks like this of states? Okay. What about this? Okay. All right, so if you did think about this for a little bit now, let's see if you understood. So I have to choose a starting state that's dictated by my pi vector, right? Oops, I don't want to do that. And basically, I have to start in state A. There's no chance that I'll start in state B. So I start here in state A, and I roll the die, and with 90% probability, I stay in state A. And with 10% probability, I switch to state B. So for example, I might come over here in state A. I roll the dice, and I'll be in A, and then I know the first state was A. And I might roll the die, and I'll stay in state 8 for a while. And eventually, I'll get lucky, if you want to call it that, take this rare event of 0.1, and I'll switch to state B. Now I'm over here. And now, with probability 1, I will stay in state B. There's no probability I go back to A. So I will just now be, for as long as I let this Markov model run, you'll just keep adding B's down there. Second challenge then, here. I have 4 states, A, B, C, D, okay, and my initial probabilities are simple, they're equifrequent. Okay, so what happens now in this Markov model? Okay, just give me examples of how this would look. Okay, it's really simple, I think. Okay, third example, this guy. The probability for A to A is 0.5. We're going to make that 0.5 for all of them. And then the other transition probabilities are 0.15, 0.15, and 0.3. It doesn't matter really how it's assigned, but it's basically the same for all the other nodes. They're basically, what I'm saying here is that all the transition probabilities overall have non-zero probabilities. And my initial probabilities are equifrequent. The question here I want you to ask is there any sequence of states that can't be produced by this Markov model? Here's an example of one sequence of states that can be. Okay, I could produce A, dot, 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 dot. And that would just be staying in state A the whole time. It might not be the most likely thing to occur, but yeah, it could occur. Is there some sequence of states that's not possible, though? Okay, so here's a challenge for a little bit of practice for you. So create a Markov model that produces the following sequence. Now this is one example, right? But it's basically, it's not infinite, what I mean to say is, but it just continues on the same way. A, B, A, B, A, B, A, B, right? So try to spell out what that Markov model will look like. And here's another one. Create a Markov model that can produce the following sequences: AE, BE, CE, or DE. Okay? Now, if it was to keep running, there would be more examples, but of A, E, B, E, C, D. Okay. Um, and I don't know there's no particular um, A to E, B, E, C, E, D, E. No one is more frequent than the other ones are. So there's little challenges for you to make sure you understand the concept of a Markov model. Okay, so now what we're going to look at for the remainder of today and in next class is the gene finding problem. So here we're given, and this is a simplification. At the end of next class, I'll show you some more industrial strength software that's used for both eukaryotic and prokaryotic gene finding. But it's important to sharpen our teeth on this very fundamental concept of Markov models and hidden Markov models in computational biology. So for simplicity here, we're going to assume it's a linear chromosome. It doesn't affect anything. So we have a stretch from the first to the last nucleotide here. And we're just simplicity here also is we're going to focus on one chromosome. If you had multiple chromosomes, you would just repeat what we're going to talk about. In general here, we don't know where the genes are. We might know where some are, but we generally don't know where they are. And our goal is to find them. So if this is my chromosome here in black, then I could imagine that there's maybe 3 genes scattered across that chromosome and separated by non-coding regions. Now, prokaryotes may not have very big non-coding regions, if any. Eukaryotes might have massive intergenic regions. Let's not worry about too much, but in our model here, we're going to assume that there is non-coding regions between the genes. Okay. Now, for simplicity of exposition, we're going to assume that the genes are really simple too. There's no introns to worry about, no regulatory elements, etc. I mean, of course, all of these extra considerations about introns, exons, structure of genes, UTRs and promoters, all sorts of things, enhancers, etc., do impact non-coding RNAs. I mean, the list goes on and on and on, but retrotransposons, I'll stop now. But there's a lot of things that do need to be considered, but the fundamental model that sits underneath that software still looks very much like what we're going to talk about here. So in essence, what we want to do is we want to basically walk from the beginning of the chromosome to the end of the chromosome, okay? And as we walk along, you can imagine each step is one nucleotide. You want to declare whether you believe that that nucleotide is a non-coding nucleotide, in other words, it's not participating in a gene, or whether you're standing in a position where it is a nucleotide that's within an exon or a gene, right? So let's just say gene and not worry about introns, etc. So we're just separating here between non-coding regions and the 3 cartoon coding regions in this figure. So this is a very simple model, right? We all agree. But let's solve it using something called a hidden Markov model. And hidden Markov models, not too surprisingly, build off of Markov models.